in the world. I now request the speaker to begin. Thank you. Mr. Reddy, you can start now.
Yes. Yeah, thank you, ma'am. Yes, on behalf of agricultureinformation.com, I would like to invite or welcome all the participants who are listening over to my guest lecture. Yes, we can see here rice is one of the most important staple food crop for more than 1 billion of people in the world because it is grown majorly in the Asian countries, and like most of the Asian countries feed on this rice as a staple food crop. And day to day, we can see this rice has been challenged by many biotic stresses such as bacterial pathogens, viral pathogens, and the fungal pathogens, and they are known to cause enumerative disease losses. Among these bacterial pathogens, if you see, look into these bacterial pathogens, there are 3 bacterial pathogens that are reported to cause diseases in this rice. These include bacterial leaf streak, bacterial panicle blight, and bacterial leaf blight. And among these bacterial blight, an important disease is the bacterial blight, which is caused by Xanthomonas. It is very prevalent and destructive disease in rice all over the world, because we can see the data here. Till to date, there are more than 46 blight resistant genes that have been developed by the breeders, have been incorporated into the rice, but still this bacteria is able to break that resistance because there is a continuous evolution of these Xanthomonas species all over the world. See, over 30 races of these Xanthomonas have been developed still. So, in coming into the modern context of error, there are some of the editing technologies, unlike the breeding programs, where these editing technologies can directly target this specific gene in the plant that where this bacteria can't cause infection in the plant. Here we will be discussing more on the genome editing tools and what is the current scenario in different countries compared to our country, India, and how whether it is feasible in India and what are the regulations that have been made in India. Yeah, coming into the topic today, I would like to speak more on the genome editing and the current scenario of genome edited crops. And myself, Namburi Karunakar Reddy from University of Agricultural Sciences, GKVK, Bangalore, pursuing third PhD. This is an outlook of genome edited crops where it is grown throughout the world. And coming to this greener area represents the genome edited crops not regulated as GMOs. Means we have to look into 2 concepts. What is that? Genome edited crops and the GMOs. Whether these genome edited crops falls under GMOs or not. Because there is a tight, like the regulations have been very close such that whether these genome edited crops can be classified into GMOs or not. If you see into the developed countries like United States of America, Canada, Colombia, and Brazil, Argentina, Chile, these genome edited crops are not regulated as GMOs because genome edited crops are nothing but we are editing in the plant, like we are removing 2 or 3 or a few nucleotides in the gene of a plant. But whereas as GMOs are, we are inserting the gene of other organism, means a distinct taxon into the distinct taxon of other organism. So we are bringing the gene of, suppose if we are bringing the gene of a bacteria into a like cotton where the BT has been evolved, that is a GMO crop. But these are not like BT crops. These are, we are making cuts in the plant. So these won't come under GMOs. But there are tight definitions between the countries. Definitions given by different countries. You can see in case of Canada, Colombia, Brazil, Argentina, and America, these genome edited crops are not regulated as GMOs. They have classified. And coming to the red color, we can see whereas the European countries, United Kingdom and the parts of New Zealand, they have classified these genome edited crops as GMOs. Means they have grouped these genome edited crops also under GMOs. Then there will be a strict impact on the regulation and the movement of such products. And coming to the yellow colored countries, we can see like India, Bangladesh, where our country is still the discussions are going on for these genome edited crops, whether we have to keep them in GMOs or as a separate classification we have to give for such a type of crops. Yes, sorry for the interruption. The genome-edited cropland we can see. Why the genome-edited cropland we are making here? Because the amount of these genome editing applications in different areas, it has been analyzed. And coming to our cropping area, different studies have been studied in different crops like rice, wheat, maize, whatnot, apple, food crops, economical crops like tobacco, cotton, sugarcane. But what is what they're trying to create in such crops is majorly the improving yield. Whether this improving yield can be brought about by only by the agronomic characters? No, we have to improve the characters of against the diseases and the pests also. See here, these genome-edited cropland, you can see how what are the characters that we have selected to improve the yield. Improved resistance against diseases, improved resistance against abiotic stresses like temperature and drought, which is majorly prevalent in the very dry land areas and in the red soil regions, and improved agronomic traits. This is the final agenda that we have to get in the as an output is a yield, which is a major agronomic trait, and improved quality traits and improved health related traits. Like health related traits in the such, if you're going for an editing of the crop, that editing should not result in the harm of a human or an animal which or whom it is consuming. We are consuming. Sorry, I didn't, I have muted. I think I have been going on. I will repeat this slide once again. This is a genome-edited cropland. We can see why this genome-edited cropland has been framed. Because this genome editing has been wide applications like in different areas. And coming to our cropping pattern, the amount and variety of genome editing applications have been studied in different crops and in different studies like the food crops, economical crops, majorly the cotton, sugarcane, all those things. And we are studying majorly against the yield traits because the ultimate agenda is to get more yield. So the genome editing, this technology has been used against the disease resistance, pest resistance, and also against the abiotic stresses, majorly the temperature and the drought, especially in the dryland areas and in the red soil areas. And also improved health traits, because ultimate is the animals or the humans whom we are going to consume such genome-edited crops. It should not have an impact on the health of the human or the animals. This is, we can see overview of all the crops for which the trait in which the of particular crop we have used the genome editing technology to improve the specific character or needed to be improved these specific characters. In case of banana, the banana streak virus, which is an emerging pathogen, and in case of rice, where the flowering needs to be done early because of the early rainfalls or the erratic rainfalls that we receive, such that erratic rainfalls will cause in shedding of these grains. So where if there is an early flowering in rice, we can overcome such conditions, like in case of mustard with improved flavor, because it is oil of high export value, such that if you are going to improve the flavor of it with a genome editing technology, we can fetch more price. Likewise, high lycopene in tomato, and some of the important crops like soybean, which we called as a vegetable meat, where the protein content has to be increased still, and the waxy maize hybrid, because maize hybrids are very susceptible to pest and disease. If you are able to create such kind of waxy with this kind of technology, we can resist these pests and diseases. And there are some other crops like fungi, like this grape, which is highly attacked by the downy mildew pathogen, fungal, and creating resistance against such type of disease will fetch more price for these grapes in the market, like creating high fiber and low gluten in wheat, and resistance to some of the fungal diseases will help in fetching more price for such kind of products. We can see like the products of genome editing, if we combine such kind of technologies with the new plant breeding technology, and if we look into the past, the plant breeding technology, they will take around 10 to 12 years to make a new variety. And if we combine such kind of technologies like this genome editing technologies with breeding, we can make the product to come at a very short time. And globally, many research activities employing genome editing have been going on, especially in India. If we come, this has been still in the lab condition compared to the global countries where they have been released the varieties to outside, and the people are still consuming without any effects on the animals and humans. And these are all the traits. We are thinking that the ultimate agenda is to increase the yield. So these are the multiple traits that have been designed here. If you are going to give a specific improve in the value of these traits, ultimately there will be an increase in the yield. If you see this 40 till now, the traits that have been developed for high yield through these genome editing technologies, where these agronomic values will play an important role, like the improving for the cultivars without lodging, and all these characters which are called as agronomic characters plays an important role, where the shattering losses will be very low if we create a variety without a lodging effect. And with we can see next is 35% food and feed quality. Means ultimately the next if a person is consuming any food, the feed quality should be good. In order to keep the aroma or the flavor of the food, we have to increase the feed quality. And many scientists are focusing on this area, food and feed quality. And next is the biotic stresses. If you say 23% of biotic stresses, what are the biotic stresses? Biotic stresses means microorganisms. That may be the fungi, that may be the bacteria or the fungi. Majorly the viruses, which evolve rapidly in the environment. And coming to the next is herbicide. Nowadays, without some people, farmers are going for erratic, they're applying it as a post-emergency, even though if we recommend as a pre-emergency, such that the crop will be destroyed. If we are able to create a herbicide tolerance in the plants, our crop can be protected within the when our crop is occupied entirely by the weeds. We can protect our crop. So these are the multiple traits which go on with a range of 5%, 6%, and this is the overall view of the traits that have been still employed in the genome editing technology. Yeah, these are the examples, classical examples for which the genome editing technologies have been still used. We can see in case of banana that disease resistance has been created, like for the fusarium wilt and the banana streak virus, by editing in this DM-R6 and BSV sequences of banana plant, host genome plant. These sequences will be there, and we are going to edit such sequences, and this will import the resistance against the fusarium wilt and the banana streak virus. In case of cassava, you can see food safety like cyanide-free has been created, quality wax, and the distance resistance has been created cassava through the sweet gene promoters. Means we are blocking some of the sweet genes in the cassava where the bacteria can't take the sugar from the plant, such that the bacteria can't grow or the fungi can't grow in the plant and it will die, so that disease resistance can be important. Similar in case of maize, weed resistance through striga and disease resistance has been created, and the potato, which is majorly favored by this for leaf roll viruses, potato virus Y, potato virus X, where the editing in the lines such as the elongation factor 4 will create lines such that they will impart resistance against these potato leaf roll viruses, especially in case of rice, which has been consumed more than billions of people in the world. Many traits have been designed here. We can see compared to the wheat, in the case of rice, many traits have been designed for genome. In order to edit such traits, we are going to get good higher yield. We can see for disease resistance for BLB and RHB, and food safety with low arsenic and cadmium. Means this resistance has been created with the sweet gene promoter. I have told how the sweet gene promoter will work. If we are able to, sweet gene will be present in the rice plant. If we are able to edit this sweet gene, the bacteria can't take the sweet from the plant, so that the bacteria can't multiply. As a result, disease resistance will be important. And moreover, the irrigation of water, irrigation quality of water of rice contains most of this arsenic and cadmium, which are very harmful to the human health. And these loci, OS, NRMP, OSPT, OSMP, OSPT8, LS7, LS2 are responsible for mobilization of this arsenic and cadmium into the rice plant. If you are able to edit this, these rice plant can't take this arsenic and cadmium into the plant, so that there will be less harm to the humans who are consuming the rice. And moreover, the insect resistance, BPH, many endemic areas have been reported in India, and creating such kind of resistance against this BPH will increase the yield. Moreover, the nitrogen remobilization and methane emission reduction is also a major parameter that has to be looked into the rice. But many studies have been taken place with the genome editing, but they haven't been still published. And coming to the wheat, majorly the rust are the major disease in the wheat. Where in case of plains like in Nilgiri Hills and going to the hills like Himalayan, where the rust occupy a major disease of the wheat. And they have edited the genes like LR67 and others for imparting the disease resistance. This is a classical example how the disease resistance has been imparted in sorghum. Sorghum plants can release some of the we are sowing the sorghum into the soil, and some weeds like striga seeds will be there in the soil. And this, when the sorghum plant germinates, the roots will release some of the stimulants, stimulant chemicals. They will, by the release of the stimulant chemicals, the striga weed seeds will germinate. So the weeds will grow in the grow along with the plants. If you this stimulant is been released by the sorghum is due to the LGS1 gene in the sorghum plant. If you are able to mutate this LGS1 in the sorghum plant, it won't release such kind of stimulants. If it won't release such kind of stimulants, the striga seeds, that is the weeds plants, can't germinate. This is the resistance how they have imported in sorghum against the striga. And these are the brief introduction of different genome editing tools we can see. Now from here we are going to the technical part of the session. Like it will be like all like molecular biology techniques, how the actual mechanism of this techniques and the CRISPR takes place. You'll be looking into it. There are many editing technologies like meganucleases, zinc finger nucleases, talons, and CRISPR-Cas9. Meganucleus has been created around 1990 and ZFNs during 2000. Talons came around in 2009, and the CRISPR-Cas1 is a recent one that has been a decade back, has been introduced into the labs. And the meganucleus are the large nucleases compounds like consisting of 25 to 30 nucleotides, and this will bind onto the opposite strands of the DNA, and this will break or cut this part of the region, so that gene won't be expressed. Whereas in case of zinc finger nucleases, you can see these are the artificially designed one. This circular one, they contain 3 domains that will specifically bind to 3 nucleotides. See, means each 4 are there on one side. Each will bind 3 nucleotides, means 12 nucleotides on one side, and this consists of an fork enzyme. And on the opposite strand also it consists of 4 domains. And this will bind to the 12 nucleotides, and this also consists of an fork 1 enzyme. And this enzyme starts cleaving from this side, and this enzyme cleaving from this side. So there will be a break in the strand, such that gene won't express. And talon is an improvement of this ZFN, where single tail effector binds to the single nucleotide. Similar, like it also consists of, and it will start cleaving from such sides. And a decade back, the CRISPR-Cas has been introduced, where it comes, where we are supplying the host genome with an extra RNA called as a guided RNA, where this specifically binds to one strand of the DNA and breaks it, such that creating a break in the DNA of the host. And compared to all these techniques, ZFNs and talons, here protein engineering is involved because these are all proteins. We have to engineer the proteins. Whereas in case of CRISPR-Cas, it is a guide RNA is the nucleotides. So designing of nucleotides is easy compared to the proteins. That's what here I mean to say. Customization of these ZFNs and talons requires protein engineering for editing, whereas this CRISPR-Cas requires only the nucleotides. So CRISPR-Cas is simple, more accurate, and less costly. And coming to this coming to the uses of these 4 editing technology, we can see the rapid increase in the growth of this CRISPR-Cas9 over the years. From 1996, there was a 1996 to till date 2019, there was a rapid increase in the usage of this CRISPR-Cas technology for editing crops of specific genes to improve the disease resistance for increasing yield. And here we are going to discuss majorly background on the CRISPR-Cas9, which is majorly used tool nowadays to improve the yield. And coming to the background of this CRISPR-Cas, why we are going to use this CRISPR-Cas? Because the world agriculture is threatened by biotic and abiotic stresses. Among abiotic stresses, as I have said previously, that plant viruses are difficult to control. These viruses play a major impact in the crop yields. Under these circumstances, and chemicals also won't function on many circumstances, these chemicals have failed to control these rapidly evolving viruses. So we have, with this background, we can use this CRISPR technology where it can cut the host specific genes such that the virus can't bind to the host genes, such that this virus infection can be reduced. And this CRISPR is a technology that has been taken from the background of bacteria, where an autoimmune system has been formed by bacteria on its own. So the name is CRISPR, means clustered regularly interspace palindromic repeats. Means clustered regularly. This is because the bacteria genome is very small and it consists of only one chromosome. There will be regions of 20 to 30 base pairs, and these will be repeated several times, spaced by some short spaces. Means in the between there will be some small spaces that will separate these regions, which are palindromic, means similar. And he was the first in identifying this. He found that in a bacteria that has known same bacteria growing in different environments, means in different environment in the sense in different salt concentrations, has different number of spacer regions. Why it is happening like that, he thought of. Like the restriction profile of this bacterial genome is different. Why it is happening in different salt concentrations? He started thinking of that, and he divided the spacers into like 30 and 36 base pairs. The region that was separated by this repeats was separated by the spacers. And what he hypothesis is, later he turned into a bioinformatician, and he said that these would be a short regularly spaced repeats. Means before the concept of CRISPR came, he named them as the short regularly spaced repeats, later renamed as a CRISPR. But observing the similar structure feature in many bacteria have a series of these genes upstream to the CRISPR loci. If you think this is a CRISPR loci, there are many such like genes which are present upstream of this CRISPR. Then there are many repeats. And what is the function of this? This is all we are making in the bacteria. How this technology from the bacteria came into the lab. So what are these repeats? That started hypothesizing. Are these repeats involved in gene regulation or any gene partitioning during cell cycle or any DNA repair? Means by any chance any breakage in the DNA of the host by the chemical mutagens, these repeats will take over place in the DNA repair. They started thinking. But Mojica, who started this work in his salt-tolerant bacteria, different way. He started, what is the role of the spacer instead of the repeat? Because there are repeats interspaced with spacer regions. What is the role of the spacer? He placed, we can see here, this is ORF is our gene. There are many Cas5, Cas1, Cas6, Cas, these are the proteins that cleave. Here we can see the repeat in the spacer region. See this, we can see the heads and that are separated by spaces. So addition or deletion, we can see here only wild type plus addition of one spacer, addition and deletion of spacer, means like addition of different spaces we can see. When different spacers are added to this bacterial profile, sensitivity is also changing. Means we came to know that addition or deletion of this spacer sequence from the bacteriophage means that is a virus. Because it is why we are telling the virus means, I will be explaining the latest slides. Because this virus, when causes infection, the spacer regions are taken from the virus to the bacteria by the Cas genes here. So based on the type of spacers and the number of spacers that have been taken by the bacteria from the virus, that makes the bacteria sensitive or resistant. This slide means to say that. Then what is the function of adding new spacer? Because every time we are infection takes place and adding a new spacer takes place. If spacer is in, why cas gene should be present? The question arises. I said cas genes are involved in generating of new spaces and repeats, but not in immunity itself. Because there will previously there was a Cas5, now we are calling it as Cas9, which is majorly and all over the world they are using this Cas9 protein for genome editing, which consists of two lobes, HNH and RUVC lobes. These Cas9 protein will take the spacer region from the virus when it infects the bacteria, and such these spaces will be added up into the bacterial genome. And this Cas9 later on the stage, when the bacteria infects the same bacteria second time, this Cas9 will cleave such kind of repeated spacers. Then what is cRNA? cRNA is CRISPR RNA, whether it targets or RNA or DNA in the host genome. RNA is nothing but it consists of only the exons. Means if intronic region is removed out, if your target site is the intronic region, and it will get automatically sliced out, because during the processing of mRNA during transcription, the intronic site would be sliced out. Even though if you are targeting a specific sequence, there will be no change. If you target the DNA, this insertion would abolish the interference, because the DNA will have the spacer region. If you are targeting such site, it will cause an interference in the DNA site and prove that this CRISPR RNA targets only DNA, not the RNA. But later, many NGS analysis, like next generation sequencing analysis for all these transcripts, they have read out. And one of the Nobel Prize winner for this designing guide RNA is Emmanuel Charpentier. She found one more RNA, which is the tracer RNA. What is this tracer RNA? See, you can see here, this is a CRISPR array. These are the Cas genes. And this beyond this Cas genes, there is a tracer RNA that is known to reduce the function of this Cas9. Means it enhances the function of the Cas9 in cleaving the CRISPR array. See here, wow, that's what I said. What are the so coming to the components of CRISPR? What are the three components that have been there in the CRISPR? That is a CRISPR RNA, Cas protein, and tracer RNA. Yeah, based on this concept that have been occurred in the bacteria, Jennifer Doudna, the receiver of the Nobel Prize for designing the guide RNA, she have received. The guide RNAs have played a major role in this genome editing technologies. Based on this technique, what that has been happening in the bacteria and the virus, they have employed a technique called, they have designed the guide RNA by in vitro synthesis, like from the labs, and they have introduced this guide RNA into the plant such that it is exactly opposite to the complementary strand, where this guide RNA will bind to the complementary strand and causes a cleavage in that strand such that your pathogen genome can't bind to the host plant. See here we can see, taking the concept of cRNA and tracer RNA in bacteria, they have designed a guide RNA. No, that's what it is, the guide RNA. And we are discussing about the Cas9. What is Cas9? Cas9 is a protein that is present in the host plant itself. We are not introducing from the outside into the host plant. It is present in the host plant itself. This Cas9 binds to, this is a PAM site. This is a guide RNA you are introducing. See here. And this is a red color one is your complementary strand of your host genome. This guide RNA specifically binds here. This is a PAM. PAM means a protospacer adjacent motif. If such sites are there, then only your guide RNA starts binding to the host. Then from here, your Cas protein will take over the control of cleaving. Your Cas protein exactly binds at the PAM site and starts cleaving this part of the DNA in the host, such that your coming in genome of your plant pathogen can't bind to the host genome. So there will be no infection process. And these are the functions of different Cas proteins that have been identified. Among them, I have said already that Cas9 has been majorly used in majority of the genome editing technologies. this is a function of the CRISPR-Cas9 system. We can see how exactly it works in a bacteria. See here, virus is infecting. It will release genomic DNA. Your Cas9, that is a Cas protein, will go and attach itself. There will be immune response. Upon second time infection, what happens is Cas7 is responsible in bringing this, see green, it brought this spacer region from the virus and incorporated into the host genome of the bacteria. So now Cas7 from here went and brought the virus genome. If you move a step back, this is a virus genome. This Cas7 went and brought the host virus genome and incorporated the bacterial genome. So when bacteria infects next time, what happens? This sequence similarity of the virus and this already virus sequence is present. So it can't, when it interferes with such kind of sequences, the virus can't, infection process can't take place. These technology have been manipulated, like used, or they have constructed from this immunity system of the bacteria into a like guide RNA system where that has been employed into the plants. See here the guide RNA that has been created. See here again the non-homologous end joining, such that see when your guide RNA binds here, there will be a break in the double stranded DNA. And this, however, the DNA repair mechanism takes place. Here mutation has been created and the DNA repairing mechanism has taken place. Means here the two strands will come close and attach itself. That is it. That's why it is non-homologous end joining. In case of here there is a double strand break, but here the two strands won't come close and attach. We have learned in the cell cycle how the chiasmata crossing over takes place. Here in the same way, the crossing over takes place and there will be a buildup of the nucleotides. Such way the DNA repair mechanism takes place such a way. So it is called as homology directed repair. And these are the areas for application of genome editing all over the world. Like in gene surgery, drug development, animal models, they have been using epigenetic variations. Means within the gene, the variations in the methylated compounds, and in making the some of the materials, and improving the yield food and increasing the fuel, like generating high fuel from less energy, less biomass. Here we can see like text with the help of the text or the genome application agriculture. Like among, there are many applications like variability studies, developing biotic and abiotic resistant crops like temperature, drought, and bacterial pathogen like microorganism resistant crops, like pathogen resistant crops, and also transgene integration. Means in order to improve the yield, we can also like put the nucleotides into it, but not of the other organism. If you are going to put of the other organism, it will be again a CMO crop. And also majorly this tool has been widely used for developing virus resistant varieties, because developing virus resistant varieties is not an easy task. So this technology has brought is very potential for developing virus resistant varieties. And these are the applications or the case studies till to date that have been studied by the scientists far over worldwide. Application of genome editing technology in plants. We can see how the gene knockout or editing, multiplex genome editing, means editing 2 or 3 genes at a time. We can see for herbicide resistance also, they have been, in case of maize, they have been applied for improving the herbicide resistance. And in case of rhiza sativa, against the biotic stresses. And these are some of the case studies that would depict who are there as viewers for this presentation. They can see how this can see the case studies, how they've been known to interfere in the plants and cause resistance in the plants, like CRISPR-Cas9 mediated viral interference in plants, such that developing resistance against the viruses. And also see here we can see the engineered polyvirus are the large group of the plants. Resistance, creating resistance against this such kind of polyvirus or gemnivirus is very difficult. So using of this engineering of the CRISPR-Cas9 has created resistance in the plants. Coming to some conclusions for today's topic is more than most of the heroes of this CRISPR in the seminal age, they're not willing to take the risks, because most this CRISPR work has been came out from an boy who has been the less age of 30. So I would like to say this youth has to focus more on this such kind of technologies in order to improve or still further the implementation of this genome editing, still precisely can be modified, still precise. And CRISPR-Cas can be used for targeting influence of Gemini viruses. I have said that virus and Gemini virus are difficult to control, and using of such technologies can create resistance against these viruses. And what are the future threats? Currently, no report of directly targeting or claiming of RNA viruses, because RNA viruses means nothing but RNA viruses is like a single strand, because up to now we are discovering double stranded. But how to claim these RNA viruses has been a difficult task for us today. And CRISPR-Cas9, I think so, modifications in this CRISPR-Cas can bring about the editing of such RNA viruses also. And it remains to be determined whether, because majority of the case studies that have been studied in India or in the lab, if you go to the foreign like USA, Canada, like where some case studies depicting the tomato increase in the tomato lycopene content, where they have been brought it to the field and have been into the market, the product increased lycopene content in the tomato product has been released into the USA market. So India also have to look into such kind of regulations, what needs to be done for bringing such kind of products into the market. And sessions for defensing questions. I would like to the participants to ask questions, and thank you. Yes, ma'am. Hello, madam. Hello. Hello.
Okay, Mr. Reddy, thank you for the presentation. I'll now take up some questions. Okay. In the meantime, can you please stop sharing the screen so that we can see your full video? Yeah. Okay. Thank you. So the first question is, what made you to take an interest in this field of research?
Yeah, daily we are discussing, since I said day to day the breeders have been facing many problems in developing a varieties. Like to develop a variety, they need almost like 10 to 12 years. And it is not sure that the resulting variety or the hybrid would give an increased yield. So if you are going to target directly such kind of traits that can be result in improving the yield, we can need to select this kind of technology. When coupled with breeding technology, we can achieve it at a very faster rate.
Okay. Next question is, how will this topic, genome-edited food crops, affect the Indian farmers?
Ma'am, if we see in in USA and the developed countries, the regulations have been made, because these genome-edited crops are not the GMOs. In India, there was a strong against the GMO like this brinjal, because eating of this brinjal will affect the human health. That's what I said in the starting, because these genome-edited crops are not the GMOs. Farmers can go directly for the usage of cultivation of such crops. There will be no impact on the human health, I would like to say. And encouraging such kind of policies, if policy makers encourage such kind of technologies, it will be helpful for the India to stand along with other countries, since where they are releasing their products into the world market.
Okay. And next question is, what advice will you give for other students wanting to do a PhD degree in agriculture research?
Agricultural research, if they want to focus majorly on the specific fields, it is majorly the ultimate is the product. You will be doing the extraordinary work in the lab, but if you're not able to convert that extraordinary work into a product, it is useless. So the product should be, there should be an outcome like product that will be going to the market and will have a more price. Your invention in the agriculture research should be converted into a product. That is the ultimate decision you have to take.
Okay. Now we have the last question. Please let us know a little about your career background and how you got interested in agriculture sector.
Ma'am, I was born in a farmer family in Telangana area, the recently formed state. And the Telangana area is very low with water scarcity and is very dry region compared to like our Andhra, the present state, the beside state Andhra Pradesh. So with I have born in a farmer family and grew up studying in a small school, like government school. Later up, I took up this BSc agriculture, and from the BSc agriculture, I have studied from my MSc, where I have been working on a different aspect, unlike the genome editing. This is a nanotechnology. Later, when I'm doing this nanotechnology in the MSc, I was I was keenly interested. Like this, we will call all these technologies unmanned aerial vehicles, because these are not in the control of man sometimes. So we call unmanned aerial vehicles, but not in the control of man. It is in the control of man, but it is not made from the man. It is already there in the environment. How we are able to convert into a proper technology is required, because nanotechnology is nothing but what we created. It is already there in the environment. Nanotechnology is nothing but the small size particles. So thereafter, I was thinking after in the PhD, like to work on the same pattern, on this concept like unmanned aerial vehicles. Later, which one more like technology is a genome editing was there in this unmanned aerial vehicles concept, like team area. So I got through I wrote an all India entrance exam and I got seat in the University of Agriculture's PhD. And right now, after coming over here, my guide asked me what aspects of what you want to work. And I had actually said that I would be interested to work on this genome editing, since it is a major area where many scientists among all the major scientists all along the world are working. So I took upon it, and right now I am pursuing it. Shortly, I will be submitting my thesis.
Okay, very good to know. So, okay, Mr. Reddy, I don't see any more questions from the participants. So now we have come to end of question round. On behalf of agricultureinformation.com, I like to thank Mr. Namburi Karunakar Reddy for the detailed presentation and answering all the questions. We also like to thank all the participants for joining this meeting. The meeting will now be closed.
Thank you, ma'am.
You're most welcome, sir. Thank you.